Introduction to Cells, Cytology, Part 1. Psalm 19 encourages us to examine God's handiwork in order to learn more about Him. While the vastness and majesty of God can be appreciated by gazing on expansive mountain vistas and distant galaxies, His power and attention to detail are even more clearly seen in a careful examination of the minute details He planned and executed as He created life. The cell has turned out to be far more complicated than anyone could have imagined just a century ago. What was once regarded as a box of organic compounds is now better described as a self-replicating, high-energy, extremely efficient factory. No one can delve into cellular biology without sensing that there is a God and that He has infinite power. Romans 1:20. Early evolutionary theories on how the first life arose have been revised and replaced repeatedly as scientists have had to contend with the marvelous complexity of even the simplest cell. The minute details, so often overlooked, are where God's power as creator and sustainer are most clearly seen. It is easy to become bogged down as you study cellular structures, functions, and reproduction, the topics of this chapter and the next few chapters. However, we should rejoice in the fact that such a study reveals more of God. If He is so powerful and careful in His creation of the minutiae of cells, He is no less concerned about eternal souls. Matthew 6:25-34. Truly, God is in the details. 3a. The structure of cells. Chemicals are the basic components of all matter. Atoms may combine to form molecules, and molecules can combine to form various cellular structures. When first proposed, the idea that living things were made up of cells seemed radical. As microscope technology advanced, it became evident that the cell was the basic unit of living things, and that cellular components, although often vastly different, were based on similar patterns in most cells. Just as atoms combine to form molecules with different properties, so the organic molecules of living things are organized in different ways to form different cellular structures. This chapter on cellular structure shows how the chemistry learned in Chapter 2 can be applied to both the structure and function of the many entities that compose a cell. 3.1. Objectives. Outline the events that led to the development of the cell theory. State the cell theory. Discuss the relationship between cell size and surface area. Key terms: cytology, cell theory. 3.1 Cell theory. Cytology, cytology, the study of cells, can be traced back over 300 years to the English scientist Robert Hooke. In 1665, he published Micrographia, a report of his observations using a simple compound microscope that could provide a magnification power of approximately 30 times. By contrast, a typical modern high school microscope may magnify the image of the specimen as much as 1,000 times. In observation 18 of Micrographia, Hooke described his observation of a thin piece of cork. I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb. These pores, or cells, were indeed the first microscopical pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that had made any mention of them before this. Because the neat rows of little boxes reminded him of the rows of rooms in a monastery, he named the structures cells. Later, he studied sections of carrots and ferns and described similar findings. In the specimens that Hooke studied, he saw only the walls of rather uniformly shaped boxes. Because all plant cells have sturdy cell walls, Hooke was looking at the walls surrounding dead cells. Even though scientists now know that the boxes are found in vastly different shapes, sizes, and arrangements, they are still called cells. The first person to describe living organisms under a microscope was Anton van Leeuwenhoek in 1674, an amateur lensmaker who had been intrigued after reading Hooke's Micrographia. However, the first two men who described cells were considered to be excellent observers. Image caption, Figure 3-2. Compare the complexity of this cell as seen through an electron microscope with the empty cells Hooke observed and drew. Instruments. It was not until the 19th century that the use of microscopes became more commonplace. Gradually, as microscopes improved, scientists began to realize that a cell is not empty, but is filled with minute structures. Although it had been recognized earlier, Robert Brown described the nucleus in 1833 as a special structure found in all cells. His and other studies attached significance to the role of the nucleus during cellular division and to the nuclei of egg and sperm cells. The year 1838 brought a startling and daring statement from a German botanist, Matthias Schleiden, SHLI Den. After studying much botanical material under a microscope, he stated that all plants are composed of cells. The next year, Theodor Schwann, a German physiologist, made a similar statement about animals. In 1855, Rudolf Virchow proposed that cells arise only from pre-existing cells. The observations of these scientists have been combined to form what is called the cell theory. Continued research over the years, by many scientists using more and more technically advanced instruments, confirm the validity of this theory, which is composed of three basic principles. The cell is the basic unit of all living things. Cells perform all the functions of living things. New cells come from the reproduction of existing cells. The cell as the basic unit of life. The size of most organisms does not indicate the size of its cells, but rather the number of its cells. For example, although a mouse and an elephant have cells of about the same size, their quantity of cells is quite different. Not all of an organism is made of cells. Those parts of an organism that are not composed of cells are made of materials that cells manufacture. An insect's body covering is a secretion of some of the insect's cells. Many of the materials composing bones are cellular secretions. Cell size, boxed section. How large can a single cell grow? The maximum size a cell can attain is limited by several factors. First, the message encoded in the DNA in the nucleus governs the size of the cell. Another factor is the ratio between the surface area of the cell and its volume. The nutrients a cell requires must pass from the outside of the cell through the membrane and to the inside. As the cell grows, its volume increases faster than does the surface area. At a certain point, the cell will need more nutrients than can pass through the membrane. Compare these three hypothetical cube-shaped cells. In the smallest cell, the surface area, 6 square millimeters, is 6 times as large as its volume, 1 cubic millimeter. The largest cell has a surface area, 54 square millimeters, that is only twice its volume, 27 cubic millimeters. In other words, as the cell gets larger, it's less adequate the surface area of the cell is to provide nutrients to the interior portions of the cell. 3-1 Cellular functions and processes. Nutrition, absorption, transport of dissolved substances into cells to serve as building blocks or energy sources. Digestion, enzymatic breakdown of substances to obtain building materials or energy. Internal functions. Synthesis, assembly of organic compounds from smaller units obtained from digestion, absorption, or some other synthesis reaction in the cell. Results in a cell's growth, secretion, or replacing worn-out cellular parts. Respiration, breakdown of food, usually glucose, with the release of energy. Movement, movement of the cell itself, locomotion, or movement of substances and structures inside the cell, internal movement. Irritability, ability to respond to external factors that affect the operations of the cell. In other words, the response or reaction of the cell to its environment. Release of materials. Excretion, removal of soluble waste from the cell. Egestion, elimination of non-soluble waste from the cell. Secretion, synthesis and release of substances from the cell. Continuation of existence. Homeostasis, ability to maintain a steady state in the cell. Reproduction, formation of new cells. The cell is the functional unit of life. Cells are responsible for all the functions of any living thing. Because the functions are so various and overlapping, it is difficult to describe them. The cellular functions given in Table 3-1 are more a list of the processes of life than a list of what cells do. Not all cells perform all these functions, but in order for an organism to be alive, all of these functions must be carried out. In complex organisms like human beings, some cells perform a few of these functions almost to the exclusion of other functions. The cell as the reproductive unit. For most cells, the biosynthesis of materials results in maintenance and growth of the cell. The science fiction cell that grew so large it swallowed New York, however, is not possible. When most cells reach a particular size, they either slow down their synthesis of materials, secrete certain synthesized materials, or divide. The built-in control of a cell, the DNA, can control only so much. Therefore, cell division after a limited amount of growth is essential for most cells. Cells reproduce by dividing. Cellular division results in a new organism, if the organism is unicellular, new cells, if they remain in the same organism, or a gamete, in organisms that reproduce sexually. In organisms that reproduce sexually, like an egg or sperm. Processes of cellular reproduction are discussed in Chapter 5. 3.2. Objectives. Differentiate between unicellular and multicellular organisms. Distinguish between colonial organisms and organisms containing organ systems. Key terms: unicellular organism, multicellular organism, colonial organism, tissue, organ system. 3.2. Levels of cellular organization. Whether they are one-celled organisms floating in a pond or gigantic sequoia trees, living things are composed of cells. Those organisms that are composed of just one cell are called unicellular organisms. Bacteria, as well as many protozoans, some algae, and some fungi, are unicellular. Multicellular organisms are made of many cells. Some algae and fungi are multicellular, as are all animals, humans, and plants. Some multicellular organisms are colonial. A colonial organism is a collection of similar cells living together. Virtually any of a colonial organism's cells can carry on all the life processes of the organism. Thus, if they are separated from the colony, they can grow, divide, and become a new organism. Colonial organisms can vary in complexity from what appears to be a clump of cells to a highly organized mushroom. Most multicellular organisms are made of cells that are organized into tissues. A tissue is a group of similar cells that work together to carry out a specific function. The muscle cells of the body compose tissues whose primary function is movement. The layer of cells that line your digestive tract is another example of a tissue. Many multicellular organisms have organs, which are composed of several types of tissues working together to perform a specific function. The stomach is an example of an organ. It is made of muscle tissue that churns the food, a special tissue that lines the inside of the stomach to protect it from acid, nerve tissue that directs the movement of the stomach, connective tissue that holds the organ together, and cells embedded in the stomach lining that produce acid and other enzymes. All these tissues work together to help digest food. An organ system is made up of a group of organs that work together to accomplish life functions. The digestive system includes the mouth, stomach, intestines, and other organs that work together to digest food and eliminate wastes so that the body can function properly. Evolutionary bias, box note. Sometimes unicellular or colonial organisms are referred to as first or early organisms. Use of these terms should alert the reader of evolutionary bias, implying that later, more complex organisms evolved from the early organisms. However, the Bible states that man is not created since all life was created during the week of creation. However, none of the specialized systems of tissues can survive alone. They depend on other tissues and organ systems to supply their needs. Muscles provide movement, but they depend on the digestive system to provide the energy needed for movement, the circulatory system to bring them food and oxygen, and the nervous system to tell them when to move. 3.3. Objectives. Explain the difference between eukaryotic and prokaryotic cells. Describe the structure and function of the cell membrane. Describe the difference between a cell membrane and a cell wall. List the major organelles in a cell and describe their function. describe the nucleus and its structures. Key terms: organelle, eukaryotic, prokaryotic, cell membrane, lipid bilayer, cell wall, cytoplasm, mitochondrion, ribosome, endoplasmic reticulum, Golgi apparatus, lysosome, cytoskeleton, microtubule, centrosome, microfilament, flagellum, cilium, plastid, chloroplast, vacuole, vesicle, turgor pressure, nucleus, nuclear envelope, chromatin material, nucleolus. 3.3 Cellular Anatomy. Cells are so diverse in their functions and structures that finding or describing a typical cell is impossible. While some structures are common to all cells, their form or function may vary. Other structures may be found only in a particular type of cell. As cytological techniques have improved, theories of cellular structure and function have changed, and they may change again as more information is discovered. Currently, cell anatomy can be divided into three broad categories: a boundary that encloses the cell, the enclosed area, containing various structures and molecules, the nucleus that contains DNA and other materials. Two types of cells. Cells can be easily divided into two major classifications based on the structure of the nucleus and the organelles it has. An organelle, or gunelle, is a structure inside the cell that performs special functions in the cell. Organelles can be compared to the various organs in the body. Each one performs special tasks essential for the survival of the cell. Organelles with membranes around them are called membrane-bound organelles. Those lacking membranes are termed non-membrane-bound organelles. One of the two kinds of cells is eukaryotic. Eukaryotic. Eukaryotic cells have a membrane-bound nucleus and both membrane and non-membrane-bound organelles. Humans, plants, animals, and several other organisms are composed of eukaryotic cells. Bacteria, blue-green algae, and some similar organisms are termed prokaryotic cells. These cells lack a membrane around their nuclear area and contain only non-membrane-bound organelles. Differences between these two types of organisms are so significant that eukaryotes and prokaryotes are placed into separate kingdoms. Prokaryotic organisms and their cellular structures are discussed in greater detail in Chapter 10, and the remainder of this chapter is devoted primarily to eukaryotic cells. Cell boundaries. Technically, the outer boundary of the cell, which is still part of the cell, is the cell membrane. However, many cells have structures outside their cell membrane that are not actually part of the cell. These structures, cell walls and capsules, are also discussed as part of cell boundaries. Image caption: diagram of prokaryotic and eukaryotic cell. Membrane-bound nucleus. Membrane-bound organelles. Non-membrane-bound organelles. Cytoplasm. Cell membrane. Nucleoid region. Capsule. Cell wall, not found in humans or animals. Cell membranes. Every cell, whether prokaryotic or eukaryotic, has a cell membrane, sometimes called the plasma membrane, which serves as the outermost boundary of the cell itself. The cell membrane is the structure that separates the cell from its environment. At the same time that the cell membrane is protecting the cell from the environment, it must also allow the cell to have access to the environment. Nutrients must be absorbed, wastes removed, and substances produced by the cell must be secreted. One thing that membranes have in common is that they contain phospholipids. The fact that phospholipids have hydrophobic and hydrophilic ends and do not dissolve in water gives cell membranes their basic structure. 3-2 Cellular components and their functions. Cell membrane controls movement of substances into and out of the cell. Mitochondrion transforms energy stored in sugars to usable cellular energy. Ribosome synthesizes proteins. Endoplasmic reticulum synthesizes proteins and steroids and transports materials within the cell. Golgi apparatus prepares substances to be secreted by the cell. Lysosome breaks down ingested substances, old organelles, and cytoplasmic molecules. Cytoskeleton provides structure for the cell, necessary for movement and reproduction. flagella and cilia, provide locomotion or move substances over the outer surface of the cell. Vacuole provides storage. Vesicle provides storage, may contain enzymes for specific reactions. Contractile vacuole collects and pumps water out of the cell. Nucleus stores DNA, produces RNA. Cell wall protects and supports cell. Plastid houses pigments and stores starches. Special plastids, chloroplasts, are the site of photosynthesis. Central vacuole houses pigments and stores starches. Regulates turgidity of cell. These structures are found in plant cells and some other eukaryotes, but not in human or animal cells. Water is found inside and outside the cell. The phospholipids align themselves into a lipid bilayer. This is a double layer of phospholipids with the hydrophilic ends adjacent to the water environment and the hydrophobic ends toward each other, away from the water. While phospholipids are found in all cell membranes, the exact composition of the cell membrane differs between cells. One difference is the combination of other lipids, such as cholesterol and proteins, that are embedded in the membrane. These molecules impart unique properties to the membrane. Cell walls. In addition to the cell membrane, plant cells and some similar kinds of cells are surrounded by a cell wall. The cell wall is a rigid or nearly rigid structure that is located on the outside of the cell membrane. Plant cell walls are made primarily of cellulose and other carbohydrates. They are multi-layered, providing strength and rigidity. Such strong walls would seem to make the cell an isolated island. However, this is not the case. Cell walls have pores through which the cell can communicate with other cells and take in and export substances. Capsules. Many unicellular or colonial organisms have a capsule in addition to, or occasionally in place of, a cell wall. Sometimes this covering is called a slime coat. Capsules are made of cellular secretions, such as polysaccharides and lipids, of varying thickness. Unlike cell membranes and cell walls, capsules have no structural organization. Capsules give bacterial and algal colonies their shiny appearance and slimy feel. These structures protect the organism because the material must first either dissolve the capsule or pass through it before entering the organism. Cytoplasm. The term cytoplasm, SYETUPLAZEM, refers to all of the structures and materials inside the cell membrane, excluding the nucleus. The actual fluid portion of the cytoplasm, minus the organelles, is called the cytosol. Some molecules in the cytoplasm are used to rapidly build structures to move organelles around the inside of the cell in a process called cytoplasmic streaming. In some plant cells, cytoplasmic streaming moves the organelles that carry on photosynthesis throughout the cell so that they can be positioned to take best advantage of the available light. Mitochondria, often called the powerhouse of the cell. Mitochondria, mitochondria, sing, mitochondrion, are typically bean-shaped organelles that are responsible for the reactions that transform stored chemical energy stored in sugars into usable energy for the cell. The number of mitochondria contained within a cell will vary, depending on the type of cell and its energy needs. Two membranes surround the mitochondria, a smooth outer membrane and an inner membrane that has many folds. These inner folds, or cristae, not only greatly increase the surface area inside the mitochondria, but they also contain enzymes and other proteins embedded in the membrane. Many of the chemical processes used during cellular respiration occur at these sites. The mitochondrial matrix, the fluid inside the mitochondria, also contains many enzymes needed for respiration. Depending on the usual energy demands of the cell, mitochondria may have many cristae, as in muscle cells, or they may have only a few cristae. Cell walls and plant growth. The primary cell wall is formed when a plant cell is young and still growing. Containing cellulose fibers, a jelly-like substance called pectin, and water, it is flexible enough to allow continued growth. The secondary cell wall forms when the cell has reached full size. This layer between the cell membrane and the primary cell wall contains more cellulose and is arranged in alternating layers like plywood. Once the secondary wall forms, cell growth ends. Ribosomes. A ribosome is a non-membrane-bound organelle found in both prokaryotic and eukaryotic cells. These small organelles are composed of two basic units, proteins and multiple strands of RNA. The primary function of ribosomes is to line up the amino acids during the production of proteins. Ribosomes can be either free-floating in the cytoplasm or attached to the endoplasmic reticulum. Endoplasmic reticulum. The endoplasmic reticulum, often abbreviated ER, is a system of interconnected folded membranes inside the cell. In some cells, the ER is continuous with the membranes around the nucleus. There are two types of ER contained in most cells. Smooth ER is so named because it lacks any attached ribosomes. Smooth ER is found in cells that secrete sterols and in liver cells where toxic substances are broken down. Rough ER is dotted with ribosomes and is called rough ER. Rough ER is especially common in cells that produce proteins to be secreted by the cell. The ribosomes send some of the newly formed proteins into the ER, where the protein may be further modified before it is secreted. The ER also provides a transportation network for compounds inside the cell and helps maintain the cell's shape. Golgi apparatus. In 1898, Camillo Golgi first described a cellular structure that has since been found in one form or another in most eukaryotic cells. The Golgi apparatus appears as flattened, curved, membrane-covered sacs. It is important in the final processing and packaging of many complex substances produced by the cell. Once the Golgi apparatus receives these substances, they are sealed in small sacs whose membranes appear to be made by pinching off the membrane of the Golgi apparatus as it bulges outward. The Golgi apparatus works in close association with the ER lysosomes. Lysosomes, are small, irregularly shaped membrane-bound organelles filled with digestive enzymes. They are very common in the cells of protozoans, fungi, humans, and animals. The enzymes contained in the lysosomes can digest invading bacteria and viruses or food substances. Lysosomes are also important in breaking down old or non-functional cellular structures. This process permits the recycling of materials for the formation of new organelles and cell growth. Cytoskeleton. The cytoplasm is not just an unorganized region with organelles floating about in the cytosol. Like the body, eukaryotic cells have a structural component that maintains their shape. Under an electron microscope, tiny fibers can be seen crisscrossing the inside of the cell. This microscopic system of fibers is called the cytoskeleton. Two major types of fibers compose the cytoskeleton. The microtubules are straight, hollow tubes of proteins. They help maintain the cell's shape and provide tracks for the movement of organelles and substances. The centrosome, centrosome, is a region located near the nucleus that is important in the production of microtubules. The microfilaments are thinner, solid strands of protein. They are especially important for cells such as muscle cells that contract and for cells that exhibit crawling movements. Microfilaments and microtubules, along with special proteins attached to them, form a system of scaffolding, ropes, pulleys, and engines that control the cell's shape, give it strength, and move structures around inside the cell. They also play a role in cellular locomotion. Flagella and cilia. A flagellum, flagella, is a long tubular extension of the cell membrane surrounding a special arrangement of microtubules. Flagella are often longer than the cell and usually exist singly. However, they may occur in pairs or groups of 3, 4, or 5. Cilia, cilium, have an internal organization similar to that of flagella, although they are much shorter and frequently cover the entire cell or an entire section of a cell. Cilia and flagella in eukaryotic cells beat in specific patterns. Although both of these structures are used in cellular locomotion, they can also function in a different way. Instead of moving small organisms through watery environments, they can move the environment past a cell whose position is fixed. For example, the surface of the cells that form the lining of the respiratory tract is covered with thousands of small cilia and a thin layer of mucus. As the cilia beat, Mucus is carried up and away from the lungs, along with dust and bacteria that the mucus has trapped. Plastids. Plastids are found in cells of plants, algae, and a few other organisms, but not in humans or animals. They are classified as either leucoplasts, colorless plastids used as storehouses, or chromoplasts, structures that contain pigments, and usually function in synthesis processes. Leucoplasts are found in the fleshy storage areas of plants. Potatoes, root crops, and many fruits have leucoplasts that contain starches and occasionally oils. Chromoplasts contain pigments that give the plant its coloration. The chloroplast, one of the most familiar chromoplasts, is a green organelle in which light energy is converted into organic compounds. Some chloroplasts are cup-shaped and nearly as large as the cells that hold them. Others are flat or even spiral-shaped. The most common chloroplasts are round or football-shaped. Internally, the chloroplast contains a complex system of flattened sacs called thylakoids. The thylakoids are arranged into stacks called grana, singular granum. The membranes of the grana contain the green pigment chlorophyll, which catches sunlight for the manufacture of sugar in the process of photosynthesis. Vacuoles and vesicles. Both vacuoles and vesicles are multi-purpose membrane-bound organelles, and the difference between them is often hard to discern. Although vacuoles are found in all types of cells, they are more common in plant cells. Vacuoles, vacuoles, can be compared to various household containers, different boxes, jars, and bags, that are all used to hold various substances. Vacuoles are membrane-bound organelles that contain food, water, wastes, or other materials. They tend to be stationary in the cell or to move very slowly. Vesicles, vesicles, are also membrane-bound organelles. Although there are no distinctly defined size differences between them, vesicles are usually smaller and more mobile than vacuoles. The best way to learn about the two structures is to examine some of the more common and important types. Food vacuole. Food vacuoles are formed when the cell takes in food, a process called ingestion. Lysosomes fuse with the vacuolar membrane, releasing their digestive enzymes into the vacuole. The enzymes break down the food into substances that will pass through the membrane into the cytoplasm, where they will be used by the cell. Waste vacuole. After the lysosomal enzymes have completed their task, the indigestible material that remains in the vacuole is now termed a waste vacuole. Cytoplasmic streaming moves the waste vacuoles to the inner surface of the cell membrane, where the vacuole fuses with it, pushing the wastes outside of the cell. The elimination of non-soluble waste is called egestion. Central vacuole. Most plants contain a large central vacuole. The central vacuole can be quite large and may take up to 90% of the total volume of the cell, pushing the cytoplasm and other organelles out to the thin rim of the cell. Because the cytoplasm is pressed against the cell membrane, the outward pressure exerted from outside the cell moves into the vacuole by osmosis. As the vacuole expands, pressure is exerted on the vacuole membrane, which pushes the cytoplasm and its contents against the rigid cell wall. This cellular fullness, caused by the water pressure in the central vacuole, is called turgor pressure. The difference between crisp and wilted lettuce is the result of the plant cells being turgid, crisp, or not being turgid, wilted. Contractile vacuole. Many unicellular organisms that live in water environments do not have cell walls. These cells, it seems, should burst, since water molecules quickly come through the cell membrane to equalize the concentration difference between the water environment and the cytoplasm inside the cell, osmosis. Under normal circumstances, the cell collects these water molecules in the contractile vacuoles. After reaching a certain size, the contractile vacuole fuses with the cell membrane and ejects the water. Secretion vesicle. These vesicles are usually formed by the endoplasmic reticulum, or Golgi apparatus. Vesicles transport the materials made by these structures to the cell membrane. When the vesicle membrane and the cell membrane unite, the materials are secreted. The nucleus. The nucleus is sometimes called the control center of the cell, and is usually the most prominent structure seen when observing a stained cell with a microscope. The nucleus is the site where DNA replication and RNA transcription take place. The nuclear envelope is a double membrane that completely surrounds the nucleus. In some cells, the nuclear envelope is continuous with the endoplasmic reticulum. The nuclear envelope has openings or nuclear pores, which permit the passage of material between the cytoplasm and the protein-rich fluid, the nuclear sap, inside the nuclear envelope. Contained within the nuclear envelope and the nuclear sap are the DNA and other proteins that make up the chromatin material. All of the hereditary information of the cell is stored in the DNA. The nucleolus, nucleus, is a dark staining spherical structure found in most cells. Although some cells may contain as many as 4 nucleoli, some cells may not contain any at all. The nucleolus contains large concentrations of RNA and is the site in the nucleus where ribosomes are partially assembled before passing through the nuclear pores into the cytoplasm. Facets of biology: cytoskeleton, cellular support, and movement. The cytoskeleton is an elaborate network of protein fibers inside the cell that extends from the nucleus to the cell membrane. As the skeleton portion of the name implies, the cytoskeleton gives support to the cell, much like the bony skeleton supports the body. Unlike a bony skeleton, however, the cytoskeleton is constantly being reorganized as the cell changes its shape, moves, and rearranges its cytoplasmic contents. Some components of the cytoskeleton provide for the structural integrity of the cell, others form tracks on which organelles are moved around, and still others move the cell itself. The cytoskeleton is composed of 2 primary structures, microtubules and microfilaments, each composed of different protein subunits. Thousands of protein subunits can be organized into a single microtubule or microfilament. Microtubules are the thicker of the cytoskeleton components and are very important in the organization of the cell, in cell division, and in cellular movement. They are long, somewhat stiff tubular structures that can be rapidly assembled, disassembled, and reassembled as needed by the cell. Not only do these microtubules give shape and support to the cell, but they also can provide tracks to transport organelles and other cellular substances inside the cell. Special motor proteins bind the microtubules to specific cytoplasmic components, so they can travel up and down the microtubule. In most animal and human cells, the microtubules originate from the centrosomes and extend out to the cell membrane. The centrosome is important in cellular division and is discussed in Chapter 5. Inside the centrosomes of human cells and some animal cells are 2 structures called centrioles. However, scientists are unsure of the centriole's function in the centrosome. Microtubules are also an important part of cilia and flagella. Both flagella and cilia are projections of the cell membrane that have an internal structure of microtubules. The internal structures of cilia and flagella are the same. An extension of the cell membrane covers a bundle of 9 microtubule doublets, pairs arranged in a ring, and 2 unpaired microtubules are in the middle of the ring. Although cilia and flagella are structurally similar, the movements of cilia and flagella are different. Cilia have coordinated movements similar to the rowing of oarsmen. Flagella make wave-like movements along their length, just as if a piece of rope were being shaken rapidly up and down. Microfilaments are the thinner of the cytoskeleton elements. Each microfilament is composed of 2 molecules of the protein actin twisted around each other. Like microtubules, they can be assembled and disassembled as needed by the cell, and many different kinds of proteins can bind to the microfilament, depending on the function to be performed. Some of the microfilaments are located just under the cell membrane and form a tough, resilient network that gives the cell mechanical strength and imparts shape. During cell division, discussed in Chapter 5, microfilaments help form the contractile rings that pinch the cell into 2 separate cells. Image captions. Movement of cytoplasmic contents is coordinated by special motor proteins and microtubules. The 2 basic elements of the cytoskeleton: microtubules A and microfilaments B. The 9x2 microtubule arrangement in flagella, TMA, and illustration B. Flagella beat with a whip-like motion. Cilia have a repetitive cycle that consists of a power stroke and a recovery stroke, much like someone rowing a boat. Image caption. Microfilament forming a membrane protrusion or pseudopodium. TEM of a pseudopodium. Muscle cells contain large numbers of microfilaments that are linked to filaments of another protein called myosin. When these two different fibers are activated, they slide along each other, causing the cell to contract. The coordinated movement of millions of fibers sliding along each other causes muscular movements. Microfilaments are also important in another type of cellular movement. Some unicellular organisms and special cells in multicellular organisms are able to move by crawling along. One end of the microfilament attaches to special proteins that are embedded in the cell membrane, and then more subunits are added to it. As the microfilament grows, it pushes out the leading edge of the cell membrane, forming extensions. As these lengthen, the opposite side of the cell is pulled in by contraction of the microfilaments. Using this kind of movement, some white blood cells are able to slip through blood vessel walls and crawl through body tissue to hunt down disease-producing organisms.